Okay, hello everybody. Maybe the crowd outside should start coming in, or they want to stay outside. This is a roundtable for moving MPS into practice. Really, we have a fantastic panel here for you. But first of all, let me just speak a few things. I think over the last couple of days, I would say we've seen a lot of fantastic talk about the advances in MPS, both in application and the future, including the AI and also the automation scaling up. So it's a fantastic moment for the MPS community. So what we want to talk about today is really about where we are in MPS in practice and what are the challenges so we can all solve it together, think about moving it together to the forward for broader adoption. So we got a fantastic panel here. Let me just briefly introduce them, and each one of them will come up to talk about where, from their point of view, where we are in their particular role or region, and then also talk about the challenges. And then once we gone the round table, then we will open up for open questions and discussions. So a few more are still coming in to sit down, but I'll just get started because we don't want you to be in the way for your party. So as you can see, we assembled a global panel of expertise from different stakeholders. And we have, from my left, is Dr. Ashikaga. He's with National Institute of Health Services from Japan. And second one is Rihanna David, Dr. Rihanna David from AstraZeneca in the UK, representing the pharma end users. And the third one is a unique combination, Dr. Zhongze Gu from China, and he has a company called AvaTarget, as well as he chairs the biomedical engineering, one of the top program in China, the Southeast University in Nanjing. Please give him applause. Fourth one, we also brought in a end user from Bristol Myers Squibb, Dr. Rhiannon Hartwick, from lovely San Diego, US. And then the next one is Milena Menicozzi. She's with European Commission Joint Research Center from Italy. Last but not the least, I don't think I need her introduction, but I will still do that, Dr. Tagle from National Institute of Health from the US. So as I said, next I will ask each of the panelists to come up to talk about the current practice in their region, in their role, and then talk about the challenges they see. So Dr. Ashikaga, would you like to come up?
Yes, thank you for introducing me. My name is Taka Ashikaga, and I work for National Institute of Health Sciences, not Health Services, and I also work for Japanese Center for Validation of Alternative Methods, short for JACVAM. And yes, I think JACVAM, not only JACVAM, but also other BAMs like ECVAM or ICVAM were established for standardization of in vitro method. So I believe our BAMs should have an important role of standardization of MPS, incorporating with other stakeholders. Firstly, I will introduce a major ongoing project in Japan toward the practical use of MPS. Several projects are being carried out by Japan Agency for Medical Research and Development, short for AMED, and each project consists of regulatory authorities like me, and academia, and developers and potential users. And each project has leaders, and Professor Ishida and Professor Sakai, both of them are very famous specialists in this field, and both of them are my good friends. And the ultimate goal of this project is standardization of MPS developed in Japan. Next slide. And This slide shows the structure of the project. Each project is divided into some parts. For example, program 1, device manufacturing. Program 2, model development for each organ. And program 3, standardization. NIHS or JACBAM will be closely involved in the standardization. That's it. Thank you.
Thank you very much, Saka. All right. Next one is Dr. David from AstraZeneca.
Thanks. So I've just put together a few bullet points on where we are now, as I see it, at AstraZeneca, and probably more broadly across pharma, and then also what I think we need to do next to progress the adoption of MPS. So where we are at the moment is, we have models for multiple organs, and these are either individual or can be connected up. But at least at AstraZeneca, we're working with largely healthy single organ systems. We are doing clinical translation of MPS data using quantitative systems toxicology modeling, and that's really helping us to implement these models in our preclinical safety assessment and make accurate clinical predictions for how the toxicity is going to manifest in the patient. But these MPS with the high complexity are typically low throughput, so we can't test many replicates or many conditions in these models. But what the complexity does afford us is mechanistic insight, which is really valuable. We've also made a large investment in these models. That's both in cost, but also in time. They cost a lot of money to invest in and bring those in-house or to run those studies. They also have high technical requirements, so you need technically very good lab staff to run these and get reproducible data. And at the moment, we're largely using traditional endpoints. So in terms of what's next to progress these models, I think we need these models to become what I've termed drug modality agnostic. So a lot of the time we're working with small molecules here, although we are starting to work with the newer modalities, but we need models that we can use for all of the types of modalities and thinking into the future, the new types of drugs that will come along. We need to increase the throughput of these models to allow us to test all of the conditions that we want to, and also to think about being able to move safety assessment earlier in the pipeline. Automation would be really, really helpful in improving the reproducibility of these models, as well as reducing the time of our staff. I think that real-time, non-destructive monitoring of these models is going to be really, really valuable, both in terms of the information that we can get, as well as how we can adopt these in our labs. And also high content, non-targeted data that is amenable to machine learning and AI. So typically at the moment we use endpoints that we are familiar with, but if we can generate these big data sets that we can start to probe for mechanisms and pathways that we don't yet know what to look for, but the machine learning and AI can help us with, we can probably start to really understand and get better insights into the toxicity and safety risks of our drugs. Immune competency, I think, is a really important one. A lot of the models we work with are not immune competent, although this is starting to pick up, but I think that's going to be really important, particularly for a lot of the newer modalities. And also disease relevance. So as I mentioned, particularly at AstraZeneca, we work largely with healthy models, so moving more into the disease space should further enhance the translation of these models. And then lastly, I think that we have a real opportunity with these models to combine both safety and efficacy. So rather than just looking at these models for individual applications, I think we have an opportunity to start to combine these readouts, and having them from the same system is going to be really powerful.
A few years. So next will be Dr. Gu.
Thank you, Chairman. So I'm Gu from Southeast University in China. So in fact, I come from the data medical engineering lab. It's a state-key laboratory. So the main purpose of our lab is to establish a method to build a digital twin of our body. So we have data, in fact, from the clinical imaging, and also can be from animal. But anyway, I believe one important data should come from the in vitro model, such as microphysiological systems. So that's why we started a study of MPS. In fact, we hold Chinese first MPS project. It's called organochip and its precision measurement. So from this project, we established the method to fabricate chips and also established measurement systems. As a product of the project, in fact, we have just told we have a company, a spin-off company. It's called EverTarget. The company is EverTarget. It means we want to get an avatar on a chip. So based on that chip, in fact, we've just finished a tissue-on chip, and the chip is launched to the space of Chinese space station at the end of the last year. And also we use our chip to fulfill the should be Chinese first IND for a treatment of heart disease. And also in China, we established a society. It's called Organoid and Organochip. Because the name of MPS is not so popular in China, so I changed the name to Organoid and Organochip. So by now we have, although it's not so long history, but we have members over 400 and almost 100 different university, institute, and company joined our society. So, and also I will make the announcement we have, in fact, our meeting maybe have longer history than MPS conference here, because this year we have six meetings. So it will be held in Beijing and in November. So if you like, welcome everyone attend the conference. Thank you.
So that's pretty advanced in practice in IND phase, and that's pretty impressive. Just next will be Reed, Dr. Harwick from Bristol Myers.
Hi, everyone, and yes, please refer to me as Rhiannon and I have a lot of experience on committees together and the need to distinguish. So a little bit of background so that you understand where my perspective is coming from. I'm currently a Scientific Associate Director in Discovery Toxicology at the Bristol Myers Squibb San Diego location. So what that means in broad terms is I serve as a project representative for discovery programs, but then I'm also a lab lead for a team that does MPS research as one of the aspects that they do to support programs. And I'm currently the chair for the IQ MPS affiliate on which both Rhiannon and I serve. So where we see from the BMS perspective, and both with our internal experience and also serving on various consortia and conversations with our colleagues in the field, is that our current industry usage is largely proof of concept for mechanistic insights and issue resolution. Our use in regulatory decision making remains more tentative at this standpoint. So we do have a challenge in understanding our clinical predictivity, and I will caution that utilization of human cells alone is not sufficient to increase that clinical predictivity, right? We see that with the use of Hep G2s. They didn't magically solve everything, so just having human-derived cells alone is not sufficient. We really need to make sure that the models are in vivo relevant. And we need confidence in predicting both positive and negative signals. So I have a bit more of the safety slant in the perspectives here, but it's not just about predicting the molecules that have the positive signal for safety, for toxicology, but having confidence in the lack of a signal as well. And so I really like to stress that, as a community, including both model developers and users like myself and regulators, what can we really do together to increase the confidence in these models? because this really is a community effort. So I've bucketed things into 3 different categories: the utility, the rigor and reproducibility, and translatability. And from the rigor and reproducibility standpoint, I really see as opportunities to help us move the field forward more is developing our data qualification packages that are more robust and directed towards sensitivity and specificity and beyond that proof of concept stage. Understanding and demonstrating our core organ function at the start with multiple cell donors. So that is one of the challenges that we often see in bringing models into industry, is if it's been qualified in a single donor, being able to reproduce those effects when we don't have access to that original donor. So having more confidence around multiple donor readouts. And then establishing defined readout regions for our key functional parameters for organ systems, as well as our markers of perturbation. From our utility standpoint, we really need to be diligent in clearly defining our context of use. And I have heard this mentioned quite a lot in the talks throughout the week. So I'm happy to see that we are making that more of a point going forward. And ensure that we have added value beyond our currently available in vitro models. So Rhiannon brought up the cost aspect of it. If we can get the answer that we need for a specific context of use in a simpler model, there's always going to be a lean for that. So there has to be a value add. What is the MPS allowing me to answer that I can't get in another model? And gaining consensus of both our positive and negative reference compound lists for those contexts of use, as well as clinically relevant readouts specific for the context of use. From a translatability standpoint, we do see the utility in utilizing non-clinical species MPS where appropriate. It's not always going to be feasible, so I'm not asking that every model needs to have availability in every non-clinical species. It needs to be driven by the question that we have. And being able to compare that to our historical in vivo data that many of us have within our organizations to help improve our in vitro to in vivo correlation. Because again, we may have molecules that did not move past non-clinical species because of toxicity, and therefore we would never test them in the clinic. And so there's part of the coin in terms of our predictivity that we're not actually testing because we don't have that benchmark. So was it specific to the rat? And that toxicity wasn't really relevant to the human. So that's where the non-clinical species can help us bridge that understanding. And then establishing reliable methods to help us extrapolate our in vitro exposures to in vivo for risk assessment. Because at the end of the day, it's not just for safety about the toxicity signal, but what does that really mean in the context of the whole animal exposure? And I will make a shout out because, again, this is very safety slanted here, to Dr. Hickman's comment in the opening session around where can we see a big impact for MPS in the near term with respect to animal reduction in our usage. And I think there is a lot of low-hanging fruit from a pharmacology perspective in being able to utilize these models as more human relevant and making a big dent in our animal usage there.
It's a big working list. Okay, next is Dr. Menacozzi.
You can go to the next one. Hello to everybody. So I will start introducing the organization I'm working at for the people that are not familiar. So I'm working for the European Commission Joint Research Center. So the European Commission has 5 different research facilities across Europe, and we are based in the north part of Italy. So we are part of what maybe you are more familiar with, the EURL ECVAM, the European Union Reference Laboratory for Alternative to Animal Testing. And what do we do, and who we are? So we are the Science and Knowledge Service of the European Commission, and our mission is to support the EU policies with independent evidences. So what do we do in terms of MPS, and what we think is needed to move MPS into practice? So we are working across a lot of different projects, but if I need to summarize our top priority, you can see them very clear on my slides, and are words that you should be all familiar with by now, because it's been mentioned many times in different talks throughout this conference. So the top priority are standardization, qualification, and knowledge sharing. So each of them is associated to a challenge, and sometime around standardization, there is the misconcept on what standardization really means. Standardization doesn't mean that we are going all to use the same chip, the same cellular model, but standardization is needed to have a better science. So standardization will happen at the technical level, at the biological level, and is what will enable us to compare all the systems that are out there. Then there is qualification, and this is a big word, and when we start talking about qualification, there is still a lot of confusion between qualification, validation, and seems to be a very lengthy process for which maybe some of you think that is not relevant. Instead, qualification is essential. Qualification is really the key for moving MPS forward. And what I observe throughout these days of the conferences is that many big pharma are already doing some in-house qualification. The problem there is that whatever qualification they do is not shared with the rest of the world or never reach the regulatory agency. So then we need to go that extra step. And we are collaborating a lot with the EMA, the European Medical Agency, and we try to come up with the roadmap to qualification, to help you guys with the terminology, what we mean by qualification, and then to help you submitting your method for qualification. The last point is knowledge sharing. So knowledge sharing is important. How we do it at the Joint Research Center, we do it through workshop. So for example, on Monday, we organize a pre-conference workshop on qualification framework for organ on chip, and there was a very important moment, because for the first time we were sitting all together around the same table, developer and user, regulatory agency, and only throughout this discussion you really create a synergy that will enable really the progress and the advancement of MPS. The additional way how we do it, knowledge sharing, is doing some systematic review. So we did the systematic review on 7 disease areas, looking specifically for non-animal methods that are out there. And from 2008 until 2019, to give you a number, there have been published more than 3,000 non-animal methods, and 1,000 of them are MPS based. And I guess that after 2019, there have been even more MPS based. But the problem there is, when you have such of omics of methods, if you are a PhD student or if you are a new biotech, which model are you going to use? You need to put it into the context of use. So in describing your method, it's essential that you say what do you want to use it for. So these are the key points that we would like to focus our discussion on. And if you like to know more about what we do at the Joint Research Center, feel free to download the ECVAM report that has been published at the beginning of the year. You can find it online. Thank you.
Thank you, Milena. So Dr. Tagle from NIH.
Thank you, Mingy. I think the main point that I'd like to present in terms of moving MPS into practice is really takes a village. It takes all the major stakeholders to come together to define what the issues are, what the challenges are, where the opportunities, and really emphasize the importance of being able to share as a community of investigators and developers, end users, early adopters to really come together. And that's depicted in this diagram. So I see it as three major groups: the developers and the technology suppliers, both in terms of coming to a definition and standardization of platform, fabrication, and also especially in the cell sourcing and the scaffolds. And then the next group that should really come alongside into this community would be the regulatory agencies across the globe. And that would lead to really good efforts in terms of standards development, validation, qualification. And then the last stakeholder that needs to be engaged will be the pharmaceutical industry representatives, where they can come in with their expertise, and certainly in a number of different areas, especially in the area of preclinical uses. And at the NIH, we have certainly identified major areas that we are trying to move MPS into practice, especially in the United States. I put check marks on the things that are the various programs that the NIH have put together in terms of demonstration and validation through proof of concept, that we can certainly develop MPS for toxicity and safety studies, the establishment of testing centers for building validation and confidence into this technology, and then also setting up a database center that is free and open to the public to be able to deposit their data, not just U.S. Investigators, but everyone can certainly be free to deposit their or submit their data and access the data there for use. Also, a good end use would be in terms of developing models for rare and common diseases. This is where a lot of the efficacy studies, I think, can come into play, and real practical applications can come in. Also, adoption, promoting the adoption and use by pharma in drug development. We have also set up programs on clinical trials on chips to bring it closer to precision medicine, where I think if we're going to make models that would predict human response, then we better also be prepared to incorporate diversity, population diversity, into our microphysiological systems. And that's where the clinical trials on chips comes in, with many different patients, different mutation spectrums, and various population demographics being represented. Global harmonization, and we're doing that certainly here. We're coming together as a community, exchanging ideas, exchanging resources, and hopefully partnering and collaborating with one another. We are also exposing and training the next generation scientists into this new technology, and I think and anticipate that will be a big push by the International MPS Society that should be announced by the end of this conference. And then lastly, in terms of the context of use and qualification, the NIH will be setting up by the end of this year translational centers for MPS, and they're specifically tasked to bring MPS for regulatory qualification for specific contexts of use. So thank you.
Okay, questions. So feel free, there's a microphone in front here, so feel free to come up to ask questions. But I'll open up with first question, because I'm always very, very optimistic. So can maybe some of you give us maybe a few examples of actual use cases you've seen, most effective, why? Maybe tell us whether it's preclinical, clinical. I think Dan talked about precision medicine or personalized medicine. So maybe Reed, would you like to start? The actual use cases in practice today that you're seeing.
She picked on me by singling me out.
Because she didn't join us last night. No.
I think, each organization often faces their very specific challenges that they're trying to tackle with the models, and that has a lot to do with their portfolio and modalities and things like that. But some of the things that we focus on that we're trying to bridge our understanding is around cholestatic drug-induced liver injury, species relevance for some of the bone marrow toxicities that we see in terms of sensitivity between species, or between clinical and non-clinical. And then, some of the interests that we also have around GI tox and species relevance. So I think sometimes the question between non-clinical species and human is around, just the sensitivity and helping our prediction of between the efficacious doses that we need versus what we're actually, when we're actually going to have a toxicity signal. And understanding the relevance too. And there's additional species relevance, but I'll keep it short.
So is that mostly preclinical then?
We aren't exclusively looking at non-clinical species models. Where the human models are available, we're certainly tackling them, but there is a concerted effort to build internal kind of companion models in non-clinical species, for sure.
Yep, that's great. Your question?
Uh-oh.
Hi everyone, thanks for being here and doing the session. When many of us started in the US doing these MPS models, it was under the umbrella of the program that Dan was running, and it was very specifically you're working on human systems, not animal systems. And now we're starting to hear some rumblings that guys like yourselves in the industry side would like to see some animal-based MPS as a way to sort of bridge data sets. So I'd just like to hear your thoughts on the need for animal-based MPS, and if you do feel there's a need, where should we start? The mouse, the rat, monkey, dog, all of the above?
Good question.
I will start, and then I have a feeling Rhiannon's going to want to take over for me. Also, I don't want to be the only one talking, but I absolutely appreciate the comment there. Yes, and I was once a model developer myself too, so I was right there with you, right? And now I've changed to the end user side. I think, to put it as simply as possible, if you think about it from the perspective of taking a molecule into a human MPS system, whatever organ it is, and we see a positive signal for toxicity, from an organizational standpoint, it's a lot easier to walk away when we've got that positive signal. It's a lot harder to have the confidence when you have a lack of a signal, because then we don't know, is it because of the model, or is it because we don't expect that toxicity to really happen in a human? And so I think it's more about just helping us have that confidence. We're not asking to prove the negative. I'm not going to go down that road, right? But having more confidence in that negative signal. So being able to translate the in vivo data that we have in non-clinical species to an in vitro non-clinical species chip gives us a little bit more confidence that what we see, especially from a negative side, in the human chip, we can have some confidence in it. And from a species standpoint, I mean, from the safety side, we've got the pretty standard species that we always look at. Rodent, I think more often than not, rat is used, but there are cases for mouse, always, obviously. But I would not discount the cyno and dog as well.
Yeah, I might just build on that. So I guess, yeah, AstraZeneca, we've taken an approach to really be focused on human models, and so those are predominantly what we've been developing and what we're using. I think, as Rhi has said, I think there are use cases where having animal models can help us in the confidence in the models. So we have used them on a few occasions, and that's typically where we get different signals in our species. So we'll see something in the rodent and we see an absence of an effect or a different effect in the dog. So what does that mean for the human? And so when you can make that comparison in organoids or in MPS, then if you can recapitulate that, then what you see in that human model, it gives you more confidence as to whether that will actually translate or not. But yeah, we're very much, I think, where we want to be is predicting for humans, and so those human models, I think, is really where we want to be focused, and a lot of our approach has been using clinical data. And I appreciate that, often we have not got all that clinical data, but where we do, I think that's a really useful benchmark. If we can recapitulate those clinical responses, then that should be able to give us confidence in the models just as much as if we can recapitulate the in vivo, because we know that the in vivo models aren't always the best model. That's why we're trying to develop these. So I still advocate for human models as being, the direction that we should be moving in.
I was going to go next. So in Europe, I think the approach is slightly different. So we would like to have a human relevant model. So we understand the approach, the need to gain confidence and trust, but we still think that we need a relevant model, and therefore why to build an animal MPS if you could build a human MPS system. Of course, that human MPS system has a role in a specific context of use. So depending on the question that you are asking, then you could say, okay, my model, it is or it is not relevant, is reproducible, does it answer to my question or not? Does anyone else want to comment? Yeah.
Let me just ask one more time. Maybe Liana, comment on the current MPS practice use that you say already. Then we'll open up for Q&A.
Yeah. So I mean, AstraZeneca, from a safety perspective, we've got models for a number of our target organs. So we're really trying to have models across of our target organs so that we can start to, humanize and improve our preclinical safety testing. So, we have models for cardiac, for bone marrow, for liver, for kidney. I'm probably going to forget some, but we're actively working in all of these spaces. And for a number of these, they are in routine use for decision making about candidate selection, or to understand safety risks, whether that's to understand the signals that we're getting in vivo, or in a number of cases to use those in place of those in vivo models. So yeah, we have a number of examples.
Great. When you say candidate selection, you mean in the clinical stage?
No, so that's preclinically.
Preclinical
So yeah, to try and influence that candidate selection, because this, typical with safety assessment, when you do that later, your candidate drug, whatever the risks are, they're already fixed. So if you can start to influence that, you can also help to reduce the safety.
Increase the success rate.
Exactly. Yeah, that's the idea.
Okay, next question.
Thomas Hartung. Our young conference series here is giving birth in 2 days to the International MPS Society. I'm wondering what you see on the agenda for moving MPS into practice, what the International Society can contribute to this.
Dr. Gu, would you like to start?
I think the communication is very important. So maybe MPS provide a very good place for the interaction, but I think maybe some different point for the research. So for us, we enter this society is maybe from different aspect. So I say our main purpose to do is to train our bodies. So in fact, we don't want to make the disease model or for treatment, but we really want to do a combination of the MPS and AI. But from when I go on, we find more and more relationship with the MPS and or stem cell or other biological things. So I think the communication between different research fields and different researchers is important.
Yes, I completely agree with him. And regulation about safety evaluation is very varied depending on area or industry, and the field of MPS is very, very wide. So international communication and among the different fields, communication is a key to success.
How about Reed from the IQ Consortium? Is that an international organization already, just from pharma end users' point of view? Well, MPS can do?
Yeah, IQ includes pharma companies that are global. Yeah, so it's not just U.S. based, which is why Rhiannon and I know each other and work alongside each other. But I think, one of the things that I've been really happy to see throughout the week with the conference is that the conversation is really shifted from where it was several years ago to a we effort, that we are all doing this as a community, and it's not just model developers sending the messages and then end users sitting as the receivers or regulators sitting as the receivers or vice versa. That I'm really heartened to see that the whole community is kind of coming together and saying, okay, what can we do together to actually move the field forward and all have collective greater confidence? Because I do agree, ultimately we do want to get to that human productivity standpoint, ensuring the safety of our patients from the pharma side, as well as ensuring that we're getting efficacious drugs to their hands. So.
If I can build from that, it is essential what you said, that all these different stakeholders are already talking. But to me, the next level, it would be an international effort. I think we are all facing the same problem, and doesn't make any sense that something is done in the States, something else in Europe, something else in Japan or in China. So we should be much, much more clear on the terminology, for example, or on the process of qualification internationally.
Would you like to say something?
Yes, certainly. I think certainly the International MPS Society, and thank you, Thomas, for that question, will serve as really the conduit for continued communication. I mean, it's great that we meet together finally as a community of stakeholders in this MPS World Summit, but that happens a week in a whole year. But there are many opportunities to continue the dialogue beyond the conference site, to continue to collaborate, to continue to partnership, to discuss about what the issues are, and perhaps coordinating worldwide effort or strategies. And that, I think, is effectively done through an international society. And then we also certainly need to protect and take care of where the field is heading. And so we need to continue to attract and train young scientists as practitioners in this field. And I think the society should certainly play a big role in providing monthly webinars or hands-on workshops. And I think as the commercial products grow into it and have its own market share, it would be an opportunity for the vendors to really showcase, what's the latest, because we want the latest tools in our tool belt. Right? Yeah.
Okay. I would say probably from a provider's point of view, I would say probably standardization. If we can harmonize globally, that would make it much easier for us, for this technology provider. Yeah, go ahead, Rhi.
And if I can emphasize, I do think the educational efforts for the next generation of scientists is something that we can't necessarily do on our own in sitting from the pharma side of things. So I think that is a big benefit that the societies are bringing forward to give us more expertise that we can rely upon from a hiring standpoint too. So I definitely applaud those efforts on the educational front.
One
more
point, and training and education for a regulatory agency as well, because for the first time they are facing new methods, so they are not used to it. We need to help them there.
Thanks. Next question, please. Announce your name and then your company.
Yeah, I'm Nordin Shamaki from Michigan State University. I want to have a comment and then a question. So I want to take it from where Dan started about the role of microphysiological systems might bring. So we had the space or the defect in using monolayer cultures, and then we thought MPS could do better. We had the problems and limitations with animal experiments. We thought MPS can do better. Now we have another problem, which is the practice of medicine, and based on data that we obtained on some sort of homogeneous populations, and then we apply it to others. So the rule, there's another space which is opening up for the application of MPS, which is addressing this this problem of the practice of medicine based on certain data. So we want to get data using MPS that takes into consideration the diversity in different levels, ethnic diversity, genetic diversity, population, different variables in weight, in different variables. So that's a very important space I think we have to go to with the potential MPS given. The question is, what the MPS could do as this can be tackled globally, and it needs a lot of effort. Thank you.
Yes, thank you for that question, Nuruddin. So certainly I echo your comment about the need for representation of diverse samples into our MPSs. In terms of what the actual needs are going to be, stem cell or cell banks, tissue banks that represents diverse populations, and those things that are shareable with proper consent and certainly authenticated and being able to use that reliably from experiment to experiment. So in order to do that, we need the infrastructure to create such tissue and cell bank repositories. We need the SOPs and best practices for pluripotency, for differentiation, best cell culture practices. All of those are essential for this kind of analysis. And then also we need to update essentially our ability to collect data and build more into a systems biology, omics-based approach of getting readout and not be looking in terms of histology, biochemical analysis, which takes time and tends to give you a very short snapshot of what's going on, but really looking at a system-wide view of what can be happening, which would also lend itself to more data for AI and machine learning to further enhance our capabilities to not only collect the data, but to analyze and make sense of what the data's presenting us.
So I just want to put, tomorrow there's another roundtable on diversification of the data and the cell sources. So do go to that one. Okay, may I pass it to the next one? Your name and your affiliation.
Hi, I'm Linda Griffith. I'm from MIT, and I have two related observations about and questions to the panel about things I think are missing from this meeting that could move the field forward faster. The first one is stimulated by the discussion about making animal chips, and so one big missing thing that Dan just alluded to is systems modeling. We have a lot of PK, PB kind of modeling here, but systems modeling where you're really trying to do essentially a translation from in vitro to in vivo. But that is especially interesting for this community because there's an enormous revolution now going on in species translation from animals to humans using existing multi-omic data sets. I have a colleague, Doug Laufenberger, who's getting so many invitations he can't even respond to them to talk about this with pharma because there are existing data sets, and sometimes the important data is in the 11th principal component. So these are not intuitive things, and they're new ways of taking existing data from animals and translating to humans. And I'm just going to put that there because I think that is a very important concept as we think about dumping a lot of money into animal chips. Okay, so that's a question for you guys on what you might think about that and if you've started to think about these kind of systems modeling. The other question, which is much shorter, is there's no or very little sexual dimorphism being discussed here. And I had a hallway conversation with some folks from Bayer about the need for this, particularly in hugely sexually dimorphic organs like the liver. So I'm curious what you guys think about sex dimorphism. Is it another one of those, oh my God, now we have to do twice as many experiments? Or is it an opportunity to take the 0th order difference in systems analysis and start thinking about it? So I posed two questions, the systems modeling and sex dimorphism.
Vienna?
Yeah, just on the systems modeling, so I absolutely agree. I think that's really critical. And so from an MPS perspective, so that's, we do quantitative systems toxicology modeling to take the data from the MPS. We can also take in vivo data. We can take, and translate that into something that is meaningful for the clinic. So in our MPS, we have different readouts. We have toxicity, we have cell number, but what will we see in the clinic? And I think that is really, really important for, these models in terms of adopting them, advancing them, and making them useful, because just a number of cells doesn't tell us about what we're going to see in the patient. So I really agree that's critical, and I think, also going back to Thomas Harting's question about the society, I think that's an area that we can really focus on. It's a skill, it's something that we have a shortage of. We do a lot of modeling in-house, but, we need more good modelers and modelers that can do that kind of work.
And there's industry folks like Applied Biomath, for example. Having John Burke come would be great because they start to get that kind of model. Anyway, yeah.
Yeah. So yeah, I think that's
always
a really
shortage of modelers.
Really important thing to be looking at, not just the cell-based model, but the mathematical models that go alongside that.
Sex dimorphism.
Yes. So one, I completely agree with you on the modeling aspect. It is very much needed. I think a challenge that I see with what we currently have available in-house is this is not the type of data they're used to dealing with or the type of models. And so there's not just training on the user side, but also the modeler side too, that we're missing. So agree with you, but a little extra
So
plug.
it sounds like an education opportunity to build okay, I'm noted.
Absolutely.
Noted.
And then on the sex dimorphism side, so yes, in safety we have to test both male and female, that is something that is often not brought up in many of our MPS studies, and I don't hear it talked about. Yeah, I agree with you. I don't hear it talked about within the field very much, but
next meeting,
we need it.
We need to have a session on sex dimorphism. Just putting it out there.
And it's not just liver, okay?
Believe me, I know. No, it's not just.
Okay. Anyone more? Dr. Gould, would you like to comment on those? No? Okay. All right, next. Hey, Steve.
Hi, everyone. Thanks for your time, too. Steve George. I'm at the University of California, Davis. And I think all of us have heard the word standardization and reproducibility for many years, and it's kind of a slippery slope. In my own lab, right, I could get reproducible results if I used the exact same cell line every time as a monolayer, right? And this society and this group in general is building very complex organisms, right? And I think about, using mice, and everyone uses C57 black or something like that, right? And the more reproducible our systems become and the more standard they become, potentially the less diverse and the less reflective of the human population. And I know Dan already commented about cell banks and stuff, but maybe I can focus my question a little bit on the use of cell lines, IPS-derived lines, or primary cells, and how do we sort of maintain predictability in sort of clinical trials and reproducibility and standardization in this very complex world.
Yes, Jacob has a lot of experiences about validation, reproducibility, and for the good reproducibility, I think the detailed and clear protocol SOP is essential. SOP is not a material method. SOP is more detailed, and for example, the performance standard list is also essential, and during the validation process, this method should be improved. Yes, so validation study is a key to improve the reproducibility.
Anybody about standardization without sacrificing the diversification?
Yeah, so when I think about standardization, and I think I kind of touched on that a little bit with the context of use readouts, the readouts that are applicable to specific contexts of use, right? I think about it as a range because you do expect some diversity, right? But from a safety aspect, we have to know when it's not normal anymore. So if we can establish, ranges for specific readouts, and I think liver is one that we can easily use as an example because we do have some good ranges established for like albumin, for example. Knowing when we're out of that range, and it's broad, the way that we look at it right now, is really important for helping us to know that we've really perturbed the system. So I don't see it as as being very strict, like a strict range. It needs to be broad enough to capture diversity, but still give us some, guardrails to know when we've really messed up the model,
Okay. Yeah, for diversity, I think, in fact, we have a lot of clinical trial data. So that's a plenary of data there. So I think, in one opinion, so for MPS, in fact, uses additional information together with a clinical trial. So if we add the information to the clinical trial, we can have a database for different types of variety. That's the reason. So in fact, I from this point, I joined the MPS. So we want to combine the MPS data and the clinical data together to find the variety of the bodies. Maybe that is another route to approach the diversity.
I tried to focus my question about cell lines and IPS lines and primary cells, but I didn't get anyone to bite on that one. Any thoughts or preferences? I mean, I would love to work with primary cells, but they're very hard to maintain sources of them, and everyone knows IPS lines may not be fully mature, and immortalization is problematic. Do you guys have any thoughts about how that's going to impact our field?
That's a good point. That's a challenge, isn't it, in standardizing the results?
For example, pools and some cells from each individual. Yes, pool is one of the candidates to standardize the cell.
Yeah, no, I was just going to add, I think, so we use both iPSCs and primary cells. I think it's about, some of it is about standardizing or being really consistent with your protocols and your approach. So, I guess a little bit to what Rhys said earlier, if you have strict protocols and standardized ranges, then you can work with that diversity, and with our primary cells, we like to embrace that diversity and that variability, because I think that really helps with that translation. And I guess also with the iPSCs, I think it's also about knowing and understanding the limitations of what you're working with. So there will be applications where that lack of maturity actually isn't a problem, and there will be other applications where that's not appropriate. So I think it's also about understanding, what you need your model to do to know what the appropriate cell source is for that.
Good, thanks you guys.
So Steve, I think it needs to be fit for purpose, essentially. I mean, there are opportunities to use primaries, like, PDX, patient-derived tumor cells. You can just put that directly on your MPS. But you can't have neural tissue as primaries. I haven't come across anyone who's willing to donate a brain or a piece of their brain to a source of primary. So that's where those are stem cells. So it has to be essentially designed in terms of what your fit for purpose is and the context of use that you're designing your MPS. And those resources, should become standardized from that point of view.
Yeah, I think it's a good question. Knowing going in that we know there's a lack of diversity in some cell sources. We know our limitation, the use cases already. So that's one plus. Yeah.
Thanks, you guys.
Yeah,
Two
and
more.
if I can add, I think that's also why I'm an advocate of not sticking with just that one donor that worked really well, and because you're not capturing diversity that way, right? But also with many of our models, because they're complex and we're looking at incorporating different cell types for an individual organ, even changing your combos. And so I am an advocate for that where it's appropriate as well. So it can be a mixture within a given model as well. It doesn't have to be all primary or all iPSC. I've definitely worked with some models where it's a mixture of both in some respects. And it's just driven by the question that you're asking, the availability sometimes for that given organ system. So, yeah.
Okay, two more questions.
All right, this is Stefan
Short
Klaus
one.
from the University of Oslo, and that's more an organizatorial question. So with the now emerging organization for MPS, how does that interface with the already well-established ISSCR? And I refer in particular to work groups, et cetera, et cetera. Maybe you could could comment on that.
Elena?
I can follow up on that question. So right now at the Joint Research Center of the European Commission, we have a project where we try to engage more with organizations like ACCR. So one of the projects, for example, is standardization of stem cells or standardization of imaging. So we are definitely reaching out what is out there, the consortia that are there, and we try to work together to come up with solutions.
Last but not least.
Thank you. I'm Hanna van Heren from the Microfluidics Association. I'm an engineer by training, so I look at the world from an engineering point of view. So the hardware issues, is that something that plays a role also in the standardization, the hardware? And so what? Because I talk to the suppliers, the supply chain, and they say the big companies should say what we should standardize. And I haven't heard it now, so what's your opinion on that, the hardware standardization?
So big companies, I think I know who it is. Go ahead. Anybody want to comment?
Yeah, I mean, I think there is definitely a role for standardization in the hardware. As to whether we can set what those need to be, I don't know. I think you know, when you have a chip, I guess that there needs to be that standardization so that when we bring that in, we know that it does what it says it can do. So that the flow rate is as it says, that the diameters, etc. So, but yeah, I think you know that's yeah. So I think there is definitely a role to standardize that so that you know we know that when we have those, they are. The specifications are as described, I guess.
Okay, anybody else? Nope. So I'll just ask one final question, because before your macro party, can each of you just comment quickly on what you see in MPS 5 years from now? What do you see? Because with FDA's Gen 2.0 in December last year, really there's a big momentum from all the stakeholders. What do you see to come for MPS in 5 years from now? Ashikawa.
Yes, I think I'm very optimistic because technology advance, speed of improving is very high. So I think after 5 years, some models is available for practical use.
Okay, great. And Rihanna?
Yeah, I mean, I hope that we see, you know, a mindset change internally within our industry to, you know, use these models and submit the data, and also some developments in terms of throughput, automation, and other endpoints that can really leverage the, you know, the complexity and the value of these models.
And AI, yes.
Yes.
Dr. Gu.
I think the MPS is high throughput and can produce data quickly. So it should be a very convenient data provides method, especially for AI training. So I believe, because we are starting the project, believe it will be the data factory for the future AI training.
Yep. Ree?
I think coming out of the FDA Modernization Act 2.0 is the renewed focus for more directed, to have greater direction together. So I see alignment coming forward now, because we're actually talking about it more together, not just within pharma and between pharma users, but pharma users, model developers, and regulators. And that's really what's been, I think, missing for a little while. So I see that alignment coming forward, and you know, amongst my pharma colleagues, I see more of a groundswell of publishing, where we're really, I do see folks are moving towards trying to make the work that we're doing more available, not just for the model developers to have the feedback, but also it helps us have more confidence amongst ourselves in terms of, if Rhiannon has done a lot of really great work on a model, and I can see that, like, it gives us
a lot more
confidence
sharing,
to try it as well. Yeah.
Malena?
So we will have this is my dream, that we will have the first MPS model qualified by regulatory agency, and once everybody will understand the big benefit that will bring, other developer will submit for approval and qualification.
Qualified, that's a big dream, yes. Dan?
Well, I think it's not a dream. It's going to be pragmatic for me, from my point of view, because like I said, that's why we're setting up our translational centers, is to essentially have that as deliverable. And that's a 5-year activity of support. And so pragmatically, I think we are going to get there. I think what my aspiration would be for many of the alternative or new approaches methods to actually be synergized with MPS, because there are going to be opportunities, and I've seen this in this conference, of AI computational approaches being more and more used for predictive talks, and you can also use that to train, to get the training set from MPS. So there are going to be more synergies amongst all the different approaches.
My big dream, just to add, it will be a first FDA approval of using MPS data in 5 years. How's that? Well, with that, I'll end this roundtable. Thanks for participation. And don't forget, tomorrow there's another roundtable for diversification of data cell sources. Enjoy the macro party. 7 o'clock boarding in the front.